Kidneys are bean-shaped paired organs that regulate water and electrolyte balance in terrestrial vertebrates. They also remove nitrogenous wastes, usually in the form of urea, from the blood and eliminate them in the urine. The renal artery and renal vein are large blood vessels that carry blood to and from each kidney. The ureter is a long tube that transports the urine formed in the kidney to the bladder for storage prior to excretion. The outer region of the kidney is called the cortex, and the inner region is called the medulla. The nephron is a basic functional unit of the kidney. Nephrons perform the work required to maintain water and electrolyte balance. Most nephrons are located in the cortex, but some extend into the medulla. Each nephron is made up of 5 regions, each performing a different function. 1. Urine formation begins in the renal corpuscle, which filters the blood. 2. The proximal tubule actively reabsorbs electrolytes, nutrients, and water. 2/3 of the electrolytes and water filtered from the blood is returned to the blood through the proximal tubule. 3. The loop of Henle creates a steep osmotic gradient by reabsorbing water and electrolytes differentially along its U-shaped length. This gradient allows the kidney to concentrate urine. A network of blood vessels supplies the loop of Henle. The vessels return water and electrolytes to the blood without disrupting the osmotic gradient. 4. The distal tubule, like some other regions of the nephron, reabsorbs electrolytes and water, but its activity depends on the body's state of hydration and is under hormonal control. 5. Urine formation ends in the collecting duct, where more water may be reabsorbed. The collecting duct is also under hormonal control. We'll use this simplified and flattened image of a nephron to describe the functions of each of its 5 regions. The first stage of urine formation begins in the renal corpuscle with the filtration of blood. Blood entering the renal corpuscle flows into a cluster of capillaries called the glomerulus. The structure around the glomerulus is called Bowman's capsule. The glomerular capillaries have pores and are surrounded by cells with slits. These features allow molecules and ions to be filtered from the blood based on their size. The high pressure inside the capillaries pushes water and small solutes out of the pores and slits and into the surrounding capsule, where the pressure is lower. Large components, such as proteins and cells, cannot fit through the pores and slits and remain in the blood, never entering the nephron. The resulting filtrate exits the renal corpuscle. The filtrate formed in the renal corpuscle contains waste products as well as valuable nutrients, electrolytes, and water. This filtrate enters the proximal tubule, where many of the valuable substances are reabsorbed. The epithelium of the proximal tubule has microvilli, which increase the surface area available for reabsorption. Intracellular sodium is actively transported out of the cells lining the tubule, a process that uses ATP. This pumping creates a sodium ion gradient favoring the entry of sodium ions into the cell from the lumen. Sodium moves through co-transporters, which use the sodium gradient to co-transport nutrients, such as glucose, vitamins, and chloride ions, from the proximal tubule into the epithelial cell. The movement of sodium down its concentration gradient provides the energy to move the other solutes against their concentration gradients. Water follows by osmosis. The reabsorbed electrolytes, nutrients, and water enter blood vessels near the tubule and are returned to the body. 2/3 of the water and sodium chloride filtered by the renal corpuscle is returned to the body via the proximal tubule. The osmolarity of the remaining filtrate in the proximal tubule is unchanged, however, because water reabsorption is proportional to solute reabsorption. The filtrate that emerges from the proximal tubule is relatively high in wastes and low in nutrients, and it is greatly reduced in volume. This fluid enters the loop of Henle. The loop of Henle sets up a steep osmotic gradient in itself and in the surrounding tissue. The osmolarity of the filtrate inside the loop is low in the cortex and high in the medulla, and this gradient is mirrored in the surrounding tissue. The loop of Henle functions as a countercurrent exchanger. The term countercurrent refers to the flow of filtrate in opposite directions in the two limbs of the loop of Henle. The osmotic gradient would disappear if the filtrates in these adjacent tubes ran the same way. There could be no exchange of water and solutes with the surrounding tissue. Water and solutes move through the loop of Henle in the following way. The descending limb of the loop is highly permeable to water, but impermeable to solutes. Water is lost by osmosis as it moves from a region of lower solute concentration inside the tubule to a region of higher solute concentration in the surrounding tissue. The fluid inside the tubule therefore becomes more concentrated as it flows deeper into the medulla. After the fluid rounds the bend, it enters the thin ascending limb, which is highly permeable to sodium and chloride ions and moderately permeable to urea. These solutes exit passively, following their concentration gradients. This part of the loop is impermeable to water, however. In the thick ascending limb, sodium and chloride ions are actively pumped out of the tubule. Therefore, as the fluid moves up the tubule, it again becomes less concentrated. The loop of Henle is an example of a positive feedback system, in which the product of a process causes the process to occur more. The positive feedback works like this. 1. The thick ascending limb pumps solutes into the surrounding tissue. 2. The high concentration of solutes in the surrounding tissue causes water to exit the descending limb by osmosis, leaving a hypertonic fluid inside the tubule. 3. This hypertonic fluid then loses solutes passively to the surrounding tissue, thus completing the positive feedback loop. What happens to the water and salt that move out of the loop? They quickly diffuse into a blood vessel that runs along the loop and returns the water and salt to the rest of the body. This return is especially important for the water that diffuses out of the descending loop, which would otherwise dilute the solutes in the kidney. The processes that we have discussed so far, filtration, reabsorption, and the establishment of an osmotic gradient, are for the most part not subject to hormonal regulation. The major solutes in the filtrate that exits the loop of Henle are urea and other wastes, and this fluid is fairly constant in composition over time. The filtrate then enters the distal tubule, where electrolytes and water are reabsorbed. Hormones control the amount of electrolytes and water reabsorbed by this part of the kidney. Hormones also control the concentration of the resulting urine within the collecting duct. If sodium levels in the blood are low, the hormone aldosterone is released. Aldosterone stimulates the reabsorption of sodium and chloride from the distal tubule into the surrounding tissue. Water follows by osmosis, thus decreasing the volume of water in the urine. Under conditions of dehydration, antidiuretic hormone, or ADH, is released. ADH stimulates both the distal tubule and collecting duct to become highly permeable to water. Water is reabsorbed into the surrounding tissue rather than being lost as urine, thus decreasing the volume of water in the urine. Individuals with diabetes insipidus produce little or no ADH. In the absence of ADH, the distal tubule and collecting duct are relatively impermeable to water. The lack of ADH results in the formation of a copious amount of urine that is low in solutes. As a unit, the nephron is remarkably effective in regulating water and electrolyte balance and maintaining homeostasis.